In this section of our notes, we're going to be talking about how we can figure out the IV and the DV for research experiments just by looking at a table and looking at a graph, okay? So one thing to note is the IV is always going to be listed before the DV. It would actually be very confusing if the DV was listed before the IV in a table. So when I look at this table here, it looks like I have distance from the light. So we're going to take a plant, we're going to put it different distances from the light. And I'd say we're going to do 3 different plants, different distances, and we're going to put a 4th plant where there's no light. And every week, we're going to take our plants and we're going to count the number of leaves that's on each, okay? So I'm varying the distance from the light. That is the IV. So we'd write that whole thing as distance from the light. And remember, DV, think of D for data. It's the numbers that you're going to collect at the end of the period of your experiment or at the end of a certain amount of time. So every week I'm going to count and I'm going to record the data. So these numbers, these data points that I'm putting here, they represent the DV. So this is 4 is the number of leaves on a plant that was this distance from the light, okay? So my data we're collecting would be the number of leaves, okay? So number of leaves would be my dependent variable. So see how the IV and what we've changed is coming first, and then what we are going to count and record comes second. We'll have a place in our table for all these numbers to be recorded as we do the experiment. All right, constants. Let's take about, just a review, let's talk about what we need to keep constant between these 4 different plants. Otherwise, it would be an unfair test or an invalid test. So one thing that we would have to keep the same would be the type of plant, because they can produce different amounts of leaves. The amount of water on each plant needs to be the same. The age of the plant, I can go on and on. The size of the pot, the quality of the soil, if there's any fertilizer we put on, we need to make it the same, okay? All right, out of these 4 setups, which one is our control group? Our control group would be this scenario right here, okay? So the control group would be plant in the dark or no light, because it shouldn't be growing if it's in the dark. And if it is growing, then that means that something else is affecting our results that shouldn't be. We can't really trust our results here. It also gives us some data that we can compare to these other plants to see if they are significantly producing more leaves because they're in different amounts of light. All right, something else that whenever we go to make our tables, make sure that if we have numbers that are a measurement, that there is a unit involved. So I have distance from the light. Well, we don't really know, is this 5 miles? Is this 5 inches? Is it 5 feet? Or 5 meters, okay? So if you put the units at the top, then that means that everything that comes underneath that, that's the units that you use. This is 5 meters, 10 meters, 15 meters. Or you could write the unit just right behind each number, okay? But you don't have to if you put your unit up there at the top of your column. All right, let's take a look at the next one. So IV will always be listed first, and your DV comes second. So it looks like we have color of light, and see how they're listing the colors of light. So color of light would be my IV, okay? And then my DV would be the height of the plant. Remember, data, D for DV, D for data. I'm going to have to record these data points down, count and record. So this is 5 is the height of my plant, so it is my dependent variable. Let's look down here. Some exceptions is, IV comes first, DV comes second, but I don't want you to think that the very first column is always going to be the IV, because bottle label is not what's important. The fact that bottle A has this much light, and then we decided to expose B to this much light. So we are changing the amount of light we want those bottles exposed to. So if I asked you, what is the IV? Don't say bottle. The IV is the light intensity. And then it looks like what comes after these numbers that we're recording here is the pH. We're looking to see how the pH will change, and then we take the final measurements of the pH. So I would say the final pH would be what we would consider our dependent variable. Same situation down here. It says yeast strain. So yeast is a fungus. So we have a normal fungus. This is the same fungus, but it has a mutation in its DNA. And then we have another fungus with a different mutation in its DNA, okay? So what this experiment's all about is we want to see if these mutations are affecting this process that's happening in that organism, okay? So I wouldn't say the yeast strain. The yeast name is not really what's important here. What it is that we are changing the different types of mutations here to see what effect it's having. So I would say the IV would be the mutation in this case, the type of mutation or presence of mutation. And then my DV would be this whole thing right here. Even if you have no clue what these words mean, you should be able to figure out that this is my dependent variable. These are the data points that we're going to collect and write down. So that those would represent my dependent variable. All right, moving on, taking a look at graphs and how we can look at a graph and figure out what is the IV and what is the DV. Let me back this up just a little bit. Okay. All right, before we begin, let's go ahead and make sure you understand that on a graph we have two axes. This is my x-axis, and this one goes up and down is my y-axis. And how you might remember that is think of it as a long tail of a y. So this is the y and this is the x. We're going to make this easy. The DV is always going to go on the y-axis. So the DV label, whatever your DV is, you're always going to label that y-axis with that label. It might help you remember that you can take this graph, these axes, and you can turn this one into the D for DV. Okay, it's not going to look right if you write a D down here with this axis because it's not going to look like a D. Okay, so your dependent variable is always, always, always going to be on your Y in all situations. So real quick, I'm just going to look at these experiments right here. If I said for this one, what's the DV? You would say yield of potatoes. It's as simple as that. Okay, bubbles per minute is the DV. Down here, number of mites is the DV. Okay, and then respiration rate is the DV. Okay, all right, the IV is going to be the tricky part. If you have a single line or a single bar graph, your IV is going to be on the X. Okay, so you maybe have seen DV and then you turn this one into an I for the IV. It's not always the case. I'll show you the exceptions here in a little bit, but this is for simple graphs, okay? So this is a single bar graph, and what I mean by that is there's just one bar for Miracle-Gro. There's one bar for Osmocote. That's a single bar for each of these categories. Down here, you can see there's actually two bars for each species. So this is a double bar graph or a multi-bar graph, okay? So for just simple little bar graph like this, your IV, you're going to look here. So they're changing the type of fertilizer. There's Miracle-Gro and then there's Osmocote. So if I said identify the IV, you would say the type of fertilizer. Okay. Over here, if I said what is the IV, and this is just a single line graph, then here it is right here. It looks like the depth of water in which a plant is placed. So the depth at which the plant was placed equals the IV. That's what we decided. We were going to put a plant here at like 2.5 meters, and we're going to put one at this distance and so on, and then we'll count the number of bubbles. So that'd be my dependent variable. Okay. Same thing with the multi-bar graph. If you are looking at it to figure out the IV and the DV is always here, and it's always a safe bet to say that the IV would be what we're comparing here. So it looks like we are seeing the different, we're comparing different types of species of flowering plants, okay? So the flowering plant species would be what I chose to make different. All right, now I want to kind of box this one off a little bit because this one's special. This one is the exception. If you have a multi-line graph, your DV's here, but oxygen concentration is not my IV. You have to look at the key for the IV, okay? So we're going to look here. So let's write look, okay? And notice what we're changing is we're doing 3 different temperatures. So the IV equals temperature. Okay. So lines of 3, look at the key. Okay. So if you have 2 or 3 or 4 lines, you need to look at the key to figure out what that IV is. Let's fill in the notes here at the top. If you have a multi-line graph, your IV, you need to go to the key and study and figure out what are they changing between those different lines. That's your IV. All right, sometimes you guys are going to be asked to make a graph, and you have to determine whether you should be making a line graph. You're going to do that often, or a bar graph. Okay. This is how you can tell whether you should be making a bar or a line. If the categories that you're placing on the x-axis, if they must stay in order, then you should be making a line graph. If the categories that you're writing on the x-axis can be rearranged and it still makes sense, you shouldn't be making a line, you should be making a bar. Okay, so here's what I mean by that. This is a bar graph and not like a line graph connecting these two, because it doesn't matter the order that I list the fertilizer. I could have put the Osmocote label and its bar on this side and the Miracle-Gro over here. I still could draw a conclusion. So since I can put these categories in any order and it would still make sense to someone interpreting my data, then I did this as a bar graph. Let's go down here and take a look at these categories that I'm writing along the x-axis. Would it make any sense if I went 0, 5, 10, 15, 30, 20, 25, 35? If I scrambled those numbers, that wouldn't make sense. So since these numbers that we wrote here, it only makes sense if we keep them in an order, we then put data points and then we connect the lines, okay? Some things that we're going to learn is never go past your first and last data point, and never automatically connect 0 to 0, because sometimes that's not the case. You can't assume that at 0, sometimes you're going to have 0. Sometimes it's 0, 0, but if you don't take a measurement of 0, then you don't put a dot and then you don't connect back, okay? All right, if you go down here, I could have named these different plant species and I could have put them in any order. So let's say this was a petunia and this was a tulip. Did I have to list the petunias first? No, I could have done species. This species could have been the tulip and then the petunias and then the daffodils, okay? So if you can put these categories in any order and still make sense and draw conclusions, bar, not a line. Would it make any sense if I went 0, 10, 5, 15, 20 as my percents of oxygen? No. These must go in order, so I'm going to then make line graph, right? A few things I want to point out where we make mistakes is when you count up with your numbers or across your y-axis, watch your spacing, okay? So here's what I mean by that. If a distance from here to here, if that distance means 5, then that distance once again means 5 more, okay? Let's say you never took a data point at 10. You took a measurement at like 5, and then you took one like at 15. You wouldn't just go 0, 5, 15, 20, okay? You have to leave spacing there for these numbers, even if you didn't take a data point for them, okay? I'm going to show you some examples here in a second where the spacing is messed up, right? And I already mentioned this. You want to draw a straight line between each data point. Okay, so nothing curvy or anything like that. Straight line between those two, straight line between those two. And do not draw past your first and last data point. Okay, let me show the spacing real quick. Here's where you've got to be careful, is from here to here means 2, right? So that space means 2. If I go over that distance again, I should have covered 2 more. So I'm up to 4, okay? And then here to here is 6, and here to here should not be 10. There should be 8 there. And even if we didn't take a data point for 8, we just don't put a data point there, This graph actually should look like this, and it looks way different because we have not plotted our data correctly, okay? So this should have been 8, right? So watch your spacing. Keep them evenly spaced as you are adding your numbers to your axis.